There's been a lot of very interesting and a very large amount of work that has been ongoing over the last several years in both academia as well as industry as it relates to cardiac bioengineering, with topics such as gene therapy, cell therapy, and 3D printing leading the way. However, interestingly, there's been very little work done in developing bioengineered acellular vessels, or HAVs, such as has been developed by Humacyte. Humacyte is really not only the world's leader, but one of the first of its kind to develop acellular vessels that can serve as a scaffold in the treatment of vascular disease. Because it's first of its kind, we've had the fortunate ability to begin clinical studies where we've used these larger diameter human acellular vessels in patients who need dialysis access, who have end-stage renal disease, or in patients who need bypass surgery in their extremities, who suffer from peripheral arterial disease or traumatic vascular injury. And the hope here is that with our ongoing research in large diameter HAVs, we can begin to push forward the envelope and create a space for a smaller diameter HAV for use in cardiovascular disease. Patients with cardiovascular disease can be treated in a multiple variety of ways, such as medical therapy or percutaneous interventions such as stents. But in so many patients who have such severe disease, surgery is the only way to treat their disease. This type of surgery requires small diameter blood vessels that are taken from the patient's own body and used to reroute the blood flow to the blocked arteries in the patient's heart. In patients who lack their natural conduit, such as the saphenous vein, or who just have such poor quality of vein that it's not feasible to be used, it would really be advantageous not only to the patient, but to the medical community if such a product existed where we could take it off the shelf and have it sized appropriately to the patient already, and then sew that graft into the patient's coronary vasculature, allowing that acellular vessel to become repopulated with the patient's own cells, allowing hopefully improved patency compared to other diseased vessels that you could take from the patient, and also allowing the vessel to last a long time and benefit the patient who's suffering from debilitating coronary disease. We hope to demonstrate long-term efficacy of these small diameter HAVs in both preclinical trials, but hopefully in first-in-man trials as well.